Welcome to the Dr. Izzy's Medical Podcast channel.
Adaptive immune system. The adaptive immune system, also referred as the acquired immune system, is a subsystem of the immune system that is composed of specialized, systemic cells and processes that eliminate pathogens or prevent their growth. The acquired immune system is one of the two main immunity strategies found in vertebrates, the other being the innate immune system. Like the innate system, the adaptive immune system includes both humoral immunity components and cell-mediated immunity components and destroys invading pathogens. Unlike the innate immune system, which is pre-programmed to react to common broad categories of pathogen, the adaptive immune system is highly specific to each particular pathogen the body has encountered. Adaptive immunity creates immunological memory after an initial response to a specific pathogen, and leads to an enhanced response to future encounters with that pathogen. Antibodies are a critical part of the adaptive immune system. Adaptive immunity can provide long-lasting protection, sometimes for the person's entire lifetime. For example, someone who recovers from measles is now protected against measles for their lifetime. In other cases it does not provide lifetime protection, as with chickenpox. This process of adaptive immunity is the basis of vaccination. The cells that carry out the adaptive immune response are white blood cells known as lymphocytes. B cells and T cells, two different types of lymphocytes, carry out the main activities, antibody responses, and cell-mediated immune response. In antibody responses, B cells are activated to secrete antibodies, which are proteins also known as immunoglobulins. Antibodies travel through the bloodstream and bind to the foreign antigen, causing it to inactivate, which does not allow the antigen to bind to the host. Antigens are any substances that elicit the adaptive immune response. Sometimes the adaptive system is unable to distinguish harmful from harmless foreign molecules. The effects of this may be hay fever, asthma, or any other allergy. In adaptive immunity, pathogen-specific receptors are acquired during the lifetime of the organism. This acquired response is called adaptive because it prepares the body's immune system for future challenges, though it can actually also be maladaptive when it results in allergies or autoimmunity. The system is highly adaptable because of two factors. First, somatic hypermutation is a process of accelerated random genetic mutations in the antibody coding genes, which allows antibodies with novel specificity to be created. Second, VDJ recombination randomly selects one variable, V, one diversity, D, and one joining, J, region for genetic recombination and discards the rest, which produces a highly unique combination of antigen receptor gene segments in each lymphocyte. This mechanism allows a small number of genetic segments to generate a vast number of different antigen receptors, which are then uniquely expressed on each individual lymphocyte. Since the gene rearrangement leads to an irreversible change in the DNA of each cell, all progeny of that cell inherit genes that encode the same receptor specificity, including the memory B cells and memory T cells that are the keys to long-lived specific immunity. Functions. Acquired immunity is triggered in vertebrates when a pathogen evades the innate immune system and 1. Generates a threshold level of antigen and 2. Generates stranger or danger signals activating dendritic cells. The major functions of the acquired immune system include recognition of specific non-self antigens in the presence of self, during the process of antigen presentation. Generation of responses that are tailored to maximally eliminate specific pathogens or pathogen-infected cells. Development of immunological memory, in which pathogens are remembered through memory B cells and memory T cells. In humans, it takes 4 to 7 days for the adaptive immune system to mount a significant response. Lymphocytes. T and B lymphocytes are the cells of the adaptive immune system. The human body has about 2 trillion lymphocytes, which are 20 to 40% of white blood cells. Their total mass is about the same as the brain or liver. The peripheral bloodstream contains only 2% of all circulating lymphocytes. The other 98% move within tissues and the lymphatic system, which includes the lymph nodes and spleen. In humans, approximately 1 to 2% of the lymphocyte pool recirculates each hour to increase the opportunity for the cells to encounter the specific pathogen and antigen that they react to. B cells and T cells are derived from the same multipotent hematopoietic stem cells, and look identical to one another until after they are activated. B cells play a large role in the humoral immune response, whereas T cells are intimately involved in cell-mediated immune responses. In all vertebrates except Agnatha, B cells and T cells are produced by stem cells in the bone marrow. T cell progenitors then migrate from the bone marrow to the thymus, where they develop further. In adults, the peripheral lymphoid organs contain a mixture of B and T cells in at least 3 stages of differentiation. Naive B and naive T cells, which have left the bone marrow or thymus and entered the lymphatic system, but have yet to encounter their matching antigen. Effector cells that have been activated by their matching antigen, and are actively involved in eliminating a pathogen. Memory cells, the survivors of past infections. Antigen presentation. Acquired immunity relies on the capacity of immune cells to distinguish between the body's own cells and unwanted invaders. The host cells express self-antigens. These antigens are different from those on the surface of bacteria or on the surface of virus-infected host cells. The acquired immune response is triggered by recognizing foreign antigen in the cellular context of an activated dendritic cell. With the exception of non-nucleated cells, all cells are capable of presenting antigen through the function of major histocompatibility complex, MHC, molecules. Some cells are specially equipped to present antigen, and are prime-naive T cells. Dendritic cells, B cells, and macrophages are equipped with special co-stimulatory ligands recognized by co-stimulatory receptors on T cells, and are termed professional antigen-presenting cells. Several T cell subgroups can be activated by professional APCs, and each type of T cell is specially equipped to deal with each unique toxin or microbial pathogen. The type of T cell activated, and the type of response generated, depends, in part, on the context in which the APC first encountered the antigen. Exogenous antigens. Dendritic cells engulf exogenous pathogens, such as bacteria, parasites or toxins in the tissues and then migrate, via chemotactic signals, to the T cell-enriched lymph nodes. During migration, dendritic cells undergo a process of maturation in which they lose most of their ability to engulf other pathogens, and develop an ability to communicate with T cells. The dendritic cell uses enzymes to chop the pathogen into smaller pieces, called antigens. In the lymph node, the dendritic cell displays these non-self antigens on its surface by coupling them to a receptor called the major histocompatibility complex, or MHC. This MHC antigen complex is recognized by T cells passing through the lymph node. Exogenous antigens are usually displayed on MHC class II molecules, which activate CD4 plus T helper cells. Endogenous antigens. Endogenous antigens are produced by intracellular bacteria and viruses replicating within a host cell. The host cell uses enzymes to digest virally associated proteins and displays these pieces on its surface to T cells by coupling them to MHC. Endogenous antigens are typically displayed on MHC class I molecules and activate CD8 plus cytotoxic T cells. With the exception of non-nucleated cells, MHC class I is expressed by all host cells. T lymphocytes. CD8 plus T lymphocytes and cytotoxicity. Cytotoxic T cells, also known as TC, killer T cell, or cytotoxic T lymphocyte are a subgroup of T cells that induce the death of cells that are infected with viruses and other pathogens, or are otherwise damaged or dysfunctional. Naive cytotoxic T cells are activated when their T cell receptor, short term as TCR, strongly interacts with a peptide-bound MHC class I molecule. This affinity depends on the type and orientation of the antigen, MHC complex, and is what keeps the CTL and infected cell bound together. Once activated, the CTL undergoes a process called clonal selection, in which it gains functions and divides rapidly to produce an army of armed effector cells. Activated CTL then travels throughout the body searching for cells that bear the unique MHC class I plus peptide. When exposed to these infected or dysfunctional somatic cells, effector CTL release perforin and granulosin, cytotoxins that form pores in the target cell's plasma membrane, allowing ions and water to flow into the infected cell, and causing it to burst or lysa. CTL release granzyme, a serine protease encapsulated in a granule that enters cells via pores to induce apoptosis. To limit extensive tissue damage during an infection, CTL activation is tightly controlled and in general requires a very strong MHC, antigen activation signal, or additional activation signals provided by helper T cells. On resolution of the infection, most effector cells die and phagocytes clear them away, but a few of these cells remain as memory cells. On a later encounter with the same antigen, these memory cells quickly differentiate into effector cells, dramatically shortening the time required to mount an effective response. Helper T cells. CD4 plus lymphocytes, also called helper T cells, are immune response mediators, and play an important role in establishing and maximizing the capabilities of the acquired immune response. These cells have no cytotoxic or phagocytic activity, and cannot kill infected cells or clear pathogens, but, in essence, manage the immune response by directing other cells to perform these tasks. Helper T cells express T cell receptors, TCR, that recognize antigen bound to class II MHC molecules. The activation of a naive helper T cell causes it to release cytokines, which influences the activity of many cell types, including the APC, which is antigen-presenting cell, that activated it. Helper T cells require a much milder activation stimulus than cytotoxic T cells. Helper T cells can provide extra signals that help activate cytotoxic cells. Th1 and Th2, helper T cell responses. Classically, two types of effector CD4 plus T helper cell responses can be induced by a professional APC, designated Th1 and Th2, each designed to eliminate different types of pathogens. The factors that dictate whether an infection triggers a Th1 or Th2 type response are not fully understood, but the response generated does play an important role in the clearance of different pathogens. The Th1 response is characterized by the production of interferon gamma, which activates the bactericidal activities of macrophages, and induces B cells to make opsonizing and complement-fixing antibodies, and leads to cell-mediated immunity. In general, Th1 responses are more effective against intracellular pathogens. The Th2 response is characterized by the release of interleukin-5, which induces eosinophils in the clearance of parasites. Th2 also produce interleukin-4, which facilitates B cell isotype switching. In general, Th2 responses are more effective against extracellular bacteria, parasites including helminths and toxins. Like cytotoxic T cells, most of the CD4 plus helper cells die on resolution of infection, with a few remaining as CD4 plus memory cells. Increasingly, there is strong evidence from mouse and human-based scientific studies of a broader diversity in CD4 plus effector T helper cell subsets. Regulatory T cells, short-term as TREG, have been identified as important negative regulators of adaptive immunity as they limit and suppress the immune system to control aberrant immune responses to self-antigens, an important mechanism in controlling the development of autoimmune diseases. Follicular helper T cells, short-term as TFH, are another distinct population of effector CD4 plus T cells that develop from naive T cells post-antigen activation. TFH cells are specialized in helping B cell humoral immunity as they are uniquely capable of migrating to follicular B cells in secondary lymphoid organs and provide them positive paracrine signals to enable the generation and recall production of high-quality affinity-matured antibodies. Similar to TREGs, TFH cells also play a role in immunological tolerance as an abnormal expansion of TFH cell numbers can lead to unrestricted autoreactive antibody production causing severe systemic autoimmune disorders. The relevance of CD4 plus T helper cells is highlighted during an HIV infection. HIV is able to subvert the immune system by specifically attacking the CD4 plus T cells, precisely the cells that could drive the clearance of the virus, but also the cells that drive immunity against all other pathogens encountered during an organism's lifetime. Gamma delta T cells. Gamma delta T cells possess an alternative T cell receptor, TCR, as opposed to CD4 plus and CD8 plus alpha beta T cells and share characteristics of helper T cells, cytotoxic T cells and natural killer cells. Like other unconventional T cell subsets bearing invariant TCRs, such as CD1d restricted natural killer T cells, gamma delta T cells exhibit characteristics that place them at the border between innate and acquired immunity. On one hand, gamma delta T cells may be considered a component of adaptive immunity in that they rearrange TCR genes via VDJ recombination, which also produces junctional diversity, and develop a memory phenotype. On the other hand, however, the various subsets may also be considered part of the innate immune system where a restricted TCR or NK receptors may be used as a pattern recognition receptor. For example, according to this paradigm, large numbers of V-gamma9 and V-delta-2 T cells respond within hours to common molecules produced by microbes, and highly restricted intraepithelial V-delta-1 T cells respond to stressed epithelial cells. B lymphocytes and antibody production. B cells are the major cells involved in the creation of antibodies that circulate in blood plasma and lymph, known as humoral immunity. Antibodies, also known as immunoglobulin, Ig, are large Y-shaped proteins used by the immune system to identify and neutralize foreign objects. In mammals, there are 5 types of antibody: IgA, IgD, IgE, IgG, and IgM, differing in biological properties. Each has evolved to handle different kinds of antigens. Upon activation, B cells produce antibodies, each of which recognize a unique antigen, and neutralizing specific pathogens. Antigen and antibody binding would cause 5 different protective mechanisms. First mechanism, agglutination, reduces number of infectious units to be dealt with. Second mechanism, activation of complement, cause inflammation and cell lysis. Third mechanism, opsonization, coating antigen with antibody enhances phagocytosis. Fourth mechanism, antibody-dependent cell-mediated cytotoxicity. Antibodies attached to target cell cause destruction by macrophages, eosinophils, and NK cells. Fifth mechanism, neutralization, blocks adhesion of bacteria and viruses to mucosa. Like the T cell, B cells express a unique B cell receptor, BCR, in this case, a membrane-bound antibody molecule. All the BCR of any one clone of B cells recognizes and binds to only one particular antigen. A critical difference between B cells and T cells is how each cell sees an antigen. T cells recognize their cognate antigen in a processed form, as a peptide in the context of an MHC molecule, whereas B cells recognize antigens in their native form. Once a B cell encounters its cognate antigen, it further differentiates into an effector cell, known as a plasma cell. Plasma cells are short-lived cells that secrete antibodies. These antibodies bind to antigens, making them easier targets for phagocytes, and trigger the complement cascade. About 10% of plasma cells survive to become long-lived antigen-specific memory B cells. Already primed to produce specific antibodies, these cells can be called upon to respond quickly if the same pathogen reinfects the host, while the host experiences few, if any, symptoms. Immunological memory. When B cells and T cells are activated, some become memory B cells and some memory T cells. Throughout the lifetime of an animal, these memory cells form a database of effective B and T lymphocytes. Upon interaction with a previously encountered antigen, the appropriate memory cells are selected and activated. In this manner, the second and subsequent exposures to an antigen produce a stronger and faster immune response. This is adaptive in the sense that the body's immune system prepares itself for future challenges, but is maladaptive, of course, if the receptors are autoimmune. Immunological memory can be in the form of either passive short-term memory or active long-term memory. Passive memory. Passive memory is usually short-term, lasting between a few days and several months. Newborn infants have had no prior exposure to microbes and are particularly vulnerable to infection. Several layers of passive protection are provided by the mother. In utero, maternal egg is transported directly across the placenta, so that, at birth, human babies have high levels of antibodies, with the same range of antigen specificities as their mother. Breast milk contains antibodies, mainly IgA, that are transferred to the gut of the infant, protecting against bacterial infections, until the newborn can synthesize its own antibodies. This is passive immunity because the fetus does not actually make any memory cells or antibodies, it only borrows them. Short-term passive immunity can also be transferred artificially from one individual to another via antibody-rich serum. Active memory. In general, active immunity is long-term and can be acquired by infection followed by B cell and T cell activation, or artificially acquired by vaccines, in a process called immunization. Immunization. Historically, infectious disease has been the leading cause of death in the human population. Over the last century, two important factors have been developed to combat their spread: sanitation and immunization. Immunization, commonly referred to as vaccination, is the deliberate induction of an immune response, and represents the single most effective manipulation of the immune system that scientists have developed. Immunizations are successful because they utilize the immune system's natural specificity as well as its inducibility. The principle behind immunization is to introduce an antigen, derived from a disease-causing organism, that stimulates the immune system to develop protective immunity against that organism, but that does not itself cause the pathogenic effects of that organism. An antigen is defined as any substance that binds to a specific antibody and elicits an adaptive immune response. Most viral vaccines are based on live-attenuated viruses, whereas many bacterial vaccines are based on acellular components of microorganisms, including harmless toxin components. Many antigens derived from acellular vaccines do not strongly induce an adaptive response, and most bacterial vaccines require the addition of adjuvants that activate the antigen-presenting cells of the innate immune system to enhance immunogenicity.
Thank you for listening Dr. Izzy's Medical Podcast Series. If you want to support this project, please subscribe the channel and like this video. Thank you.